Hello students, welcome back. In every competitive exam, you can find some questions from growth regulator or phytohormones. Okay, so phytohormones are basically when we are talking about these auxin, gibberellin, cytokinin, ethylene, and abscisic acid. So basically, you can find in every competitive exam 2 or 3 questions from that. And in this particular lecture, we'll briefly discuss about the phytohormones. What are the questions might be asked in different competitive examination from phytohormones or growth regulators. Okay, so without delay, let's dive into topic. I am Dr. Jagdish Chena, and today we are going to discuss about phytohormones. Basically, if I'll ask you what is growth regulator, doesn't only comprises these phytohormones. Okay, so growth regulators means all the factors that is affecting growth in broader aspect, if we'll say. Like internal factor or genetic makeup of that particular plant species or particular organism. What is the genetic constituents of that organism? Then you can find due to genetic engineering or breeding aspect, we can develop different high yielding varieties or high yielding plants or different biotic and abiotic stress tolerant plants. Okay, that is internal factor. Then external factor, we can have the aerial factor and soil factor, that is called edaphic factor. Aerial factor in like you can find the solar radiation, then atmospheric temperature, relative humidity, all wind speed, that is aerial factor. In soil factor, if you'll see soil physical, chemical and biological properties, like soil physical properties like soil air, soil water, then soil structure, texture, porosity, all this soil physical property. Then soil chemical properties like soil organic carbon, pH, EC, then soil nutrient status, all these are soil chemical properties. Then soil biological properties like microbes, what are the microflora, macrofauna present, what is the soil organism status in the soil, or mostly dependent on soil organic carbon, because organic carbon is the sole energy source for all these living microbes to grow. So, and microbes have potential role in mobilizing the nutrient and solubilizing the nutrients and make it available. So, microbes have various role in plant for growth and development like that. So, these are the soil. And then another aspect is management factor. Okay, management factor means how we are managing the plant or how we are managing that particular system to provide optimum resources to that plant for growth and development. So all these are the growth regulator. If we'll, in broader sense, we will tell growth regulators. These are nothing but the factors affecting the growth, like internal factor, like I discussed the genetic constituent. Then external factor, like I discussed aerial factor and edaphic factor or soil factor. Then management factor. Then in internal factor as well, this is genetic factor. Another one is this phytohormones or plant growth regulators, phytohormones. Okay, so these are phytohormones are basically the chemicals these are synthesized inside the plant in smaller quantity and they mobilize to different places. So phytohormones are basically the chemicals, they are synthesized inside the plant and they mobilize to different place to act on certain things. Okay, so there are basically 5 types of phytohormones we find inside the plant. 5 kinds of phytohormones. One is if I'll broadly group them, then they are growth promoter and growth retardant. Okay, growth promoter and growth retardant, 2 broad aspect. If we'll say the phytohormones, like 5 types of phytohormones I just told you, the auxin, gibberellin, cytokinin, abscisic acid and ethylene. So these are broadly grouped into 2 types. One type is growth promoter, another type is growth retardant. So growth promoters are auxin, gibberellin and cytokinin. These are called growth promoter. Then growth retardant, ethylene and abscisic acid. Okay, growth retardant are ethylene and abscisic acid. Growth promoter are auxin, gibberellin and cytokinin. So growth promoter are generally found in multiple form, but growth retardant, they are the actual growth regulator, because if somebody is opposing you, that is the growth regulator, that is regulating you. Suppose in case of while you are studying, suppose some distraction is there, that is regulating you, that is retarding you or obstructing you, that is the regulator of your activity. So growth retardant are basically we can say the growth regulator in short, or growth promoters are basically the present in multiple form. Okay, so right now I'll discuss one by one what are these things and some important aspects of all these phytohormones. First one is auxin. Okay, so auxin was first synthesized from, or it was first isolated from human urine. It was first isolated from human urine. Okay, then this substance, or that growth promoting substance, it was first isolated by F.W. Wendt, and he first named it auxin. F.W. Wendt was first isolated that particular substance and that he named auxin. Then what is which chemical, from which chemical it is synthesized? Actually the auxin is actually synthesized from, or the precursor, if you can say the precursor of auxin. What is the precursor of auxin? That is tryptophan. Tryptophan is the precursor of auxin. And how it is produced? See, tryptophan is the precursor of auxin. That means auxin is derived from tryptophan. Okay, so how this tryptophan produced? This is produced from Cytokinin pathway. Cytokinin pathway. See, this has a link with our herbicide part or weed management part. So this from this Cytokinin pathway tryptophan is produced. And when we apply glyphosate, you know glyphosate is a total killer. Glyphosate is a total killer. When we apply glyphosate, one enzyme it is inhibiting, that is called EPSP synthase. EPSP synthase enzyme. That enzyme, this glyphosate, actually it inhibits that enzyme, and this enzyme is helpful in Cytokinin pathway. So if we apply glyphosate, Cytokinin pathway will not happen. Okay, if Cytokinin pathway is obstructed, then tryptophan cannot be produced, and if tryptophan cannot be produced, then auxin synthesis cannot be done. So this is very important to remember. So if what it inhibits, glyphosate inhibits the Cytokinin pathway, and Cytokinin pathway, if it cannot happen, then tryptophan cannot be produced and auxin cannot be produced. Okay, so this is another important question. Next, how we can determine auxin? It is determined by using Avena curvature test. Avena curvature test. Remember these things. Avena curvature test is used for determining auxin. Okay, then some are natural auxin and some are synthesized auxin. If we'll see natural auxin, only indole 3 acetic acid, that is IAA, this is only natural auxin, indole 3 acetic acid. Then synthetic auxins are NAA, IBA, indole butyric acid, PAA, then 2,4-D. These are synthetic auxin. NAA, IBA, PAA, 2,4-D, these are called synthetic auxin. Okay, and this PAA, this is also important. This PAA is a non-indole auxin. Remember, this PAA is a non-indole auxin. Now, what are the roles of these auxins? What are the roles? Okay, auxin generally it is secreted in meristematic region or the apical region, like you can say apical bud or apical meristem. They are auxin generally synthesized. So basically it prolongs or elongates the apical meristem region, or it is elongate the growth of that particular region or the meristematic region. So if we'll see, there are specific roles of all these auxins. IBA is a rooting hormone, or it promotes rooting. Okay, IBA, indole butyric acid. Okay, it is rooting, used in rooting. 2,4-D in higher concentration, it is act as herbicide. In higher concentration, 2,4-D acts as herbicide. NAA, when we apply, that reduce the fall of leaf or immature or young stage. In young stage, if the leaf is falling or the fruit drop is there, then if we will apply NAA, that will inhibit that. So these are certain role of auxin. So these are basically what are the important aspect of auxin, or what might be the important questions from auxin we have discussed. So after auxin, let's discuss about gibberellin. Gibberellins are also called gibberellic acids. gibberellic acid. Okay, it was first synthesized from one fungus, Gibberella fujikuroi, by Kurosawa from Japan. Okay, Kurosawa, one scientist, he was from Japan, and he found first the gibberellic acid from Gibberella fujikuroi fungus, and this gibberellin, it was named by, this gibberellin term was named by Yabuta and Sumiki. Okay, Yabuta and Sumiki first named gibberellin, but it was first isolated from Gibberella fujikuroi fungus by Kurosawa in Japan. And this Gibberella fujikuroi fungus, it usually cause Becken or fully seedling in rice. Becken disease or fully seedling disease in rice. Okay, Becken or fully seedling disease in rice was caused by this Gibberella fujikuroi fungus, and from this Gibberella fujikuroi fungus, Kurosawa in Japan, he first isolated this hormone or gibberellin. Okay, now what is the precursor of gibberellin? Vernalin is the precursor. Remember, all these things are important. Vernalin. Okay, this is the precursor of gibberellin. Gibberellin generally role is it elongates the internodal elongation. It is helpful in internodal elongation. Okay, internodal elongation. Gibberellin helps in internodal elongation, and it is generally helpful in seed germination and development. Seed germination, it has immense role in seed germination. How? Because gibberellic acid, it secretes some hydrolytic enzyme like peptidase. Okay, then like peptidase or protease, then you have like lipase, then glucosidase. These are the hydrolytic enzymes secreted by gibberellin, which actually helps in seed germination. So it has immense role in seed germination, and without gibberellic acid, seed germination cannot happen. Then it inhibits the effect of dormin or abscisic acid. Gibberellic acid inhibits the effect of the growth retardant, that is abscisic acid or dormin. Abscisic acid is otherwise known as dormin. Okay, gibberellic acid has almost no real role in apical meristem and root activity. Gibberellic acid, though it promotes the seed germination and it promotes the internodal elongation, but it has no role in root activity and the apical meristematic activity. So far we have discussed Auxin and gibberellin, those are growth promoters, and now another growth promoter is there in line, that is called cytokinin. Okay, Auxin term was given by first F.W. Wen, and who termed the gibberellin? It was given by Yabuta and Sumiki, and cytokinin term was given by Litham scientist given the term cytokinin. So it is basically the precursor of cytokinin is basically adenine. Precursor of cytokinin is adenine. Cytokinin example are kinetin, zeatin. Okay, these are some example of cytokinin. Why cytokinin? Why the name cytokinin? Because it helps in cell division. Cytokinin help in cell division and elongation. That's why it is named as cytokinin by Litham. Okay. So what are the roles of cytokinin? As I discussed, the cell division. It helps in cell division. Then it inhibits the action of auxin. Okay. So what is the role of auxin? Auxin main role is apical dominance. So basically cytokinin inhibits the action of auxin and it promotes the lateral branching. Okay, auxin promotes the height. Okay, or it diminishes the growth of lateral branches, but cytokinin, it increases the lateral branches. So why? So if we are removing the apical bud, in some cases there is promotion of lateral branches or there is elongation of lateral branches. That's why due to the apical meristem, apical dominance. Okay, so due to auxin there is apical dominance. When we block the apical meristem, then there will be lateral branches. So cytokinin counteract the apical dominance of auxin and it promotes the lateral branches. And cytokinin delays senescence. See, cytokinin, it delays senescence. Okay, and that is due to Richmond-Lang effect. that is called Richmond-Lang effect. Cytokinin delays senescence, and that is called Richmond-Lang effect. Then two other phytohormones left, that is abscisic acid and ethylene. So let's discuss about abscisic acid or ABA. It is also called dormin. Abscisic acid is also called dormin. Okay, abscisic acid is also called dormin, or it is famously called anti-gibberellin. Because see, gibberellin, what it does, it inhibits dormancy. Okay, dormancy is a suspended growth phase. So gibberellin inhibits dormancy, whereas abscisic acid promotes dormancy. Okay, abscisic acid promotes dormancy. Abscisic acid is also called stress hormone. So when the plant is in stress, whether it is moisture stress or temperature stress or any type of stress, abscisic acid is always the production of abscisic acid is more, and that is why it is also called stress hormone. Okay, and what is the main mechanism of action of abscisic acid? It inhibits RNA synthesis. It inhibits RNA synthesis, and what is the precursor of abscisic acid? That is violazanthin. Violazanthin. Okay. Now next growth retardant, that is ethylene. Next, coming to ethylene. See, this is another growth retardant. Except abscisic acid, it is another growth retardant phytohormone. So ethylene is a gaseous hormone. Gaseous hormone. Okay, ethylene is a gaseous hormone, and who is the precursor of ethylene? That is methionine. Methionine is the precursor of ethylene. Ethylene increases cell permeability. Okay, and it increases the rapid maturity. So ethylene, you can in day-to-day life, we also, or fruit vendors, they also use ethylene being used to mature the or the ripening of banana or different fruit crops. They use ethylene. So these are different phytohormones we have discussed. So these are having importance. From exam point of view, you can find 2-3 questions in every competitive exam from phytohormones part. I hope you are clear all of this, like how questions may come from this phytohormone part or from this growth substances or phytohormones part, growth regulator part. So basically you have to remember from what it was isolated first, who first isolated this particular phytohormone, or who has named this phytohormone first, then what is their role, what is the role of different phytohormones, then what are the precursor of different phytohormones, like we discussed auxin, tryptophan, gibberellic acid, the mevalonic acid, or then cytokinin, the adenine, ABA, violazanthin, and ethylene, methionine. So these are the precursor of this growth regulators. So I hope all of you clear regarding all these phytohormones. So this was a basic lecture regarding phytohormones or growth regulator. Yeah, if you want to know any specific concept from agronomy or any part of agriculture, you can comment below and I'll prepare a lecture for you. So see you in the next lecture. Till then, have a great day. Bye bye.